My name is Crystal Dilworth. I'm a neuroscientist with a PhD from Caltech in Pasadena, California. It's been called a war with an invisible enemy. In 2020, we are finding out what a pandemic looks like. The world is in a fight against the novel coronavirus disease named COVID-19. We often hear the terms, scientists are working on it. But what does that mean? For this episode of VOA Tech, we wanted to know what it's like on the front lines of research during a disease outbreak. And we found out.
Powering the future. Confronting the world's challenges with cutting-edge solutions, medical breakthroughs, and high-tech discoveries. These are the stories of our planet. This is VOA Tech.
It's early morning in La Jolla, California, but the labs inside the La Jolla Institute for Immunology are already going.
Okay.
Outside, the world is dealing with the pandemic. Inside, they're doing the intricate and precise research that could one day help solve the crisis. Scientists often work in teams, each with a specific complex task. Their work has always been important. Now it's critical.
Yes, it is one of the coronavirus proteins.
Mike Norris is a postdoctoral fellow. He works in Professor Erica Ullman-Saphir's labs. They've been able to make copies of the coronavirus protein for research purposes.
Quite clearly, the vast majority of the cells have the protein we want.
That protein is part of the virus the rest of the world is trying to distance itself from. Here, it's become their main focus.
So we were just in your lab and they're doing an isolation of a protein from COVID-19. What's going on? What are you doing?
We're studying how that virus assembles. And by looking at the molecular structure by which the pieces of the virus build it together, we can understand where it's vulnerable and can begin to design antiviral drugs.
Erica Ullman-Saphir makes it sound simple. She and her research teams have solved some of the most deadly pathogens on the planet, like Ebola and Lassa. The La Jolla Institute just received a $1.7 million grant from the Bill and Melinda Gates Foundation to focus on COVID-19.
So how do you design a drug against a virus?
Viruses are remarkably simple little machines. They only have a few working parts. So their entire genome just has a few genes, which make a few molecules. And they copy those few molecules over and over again to build their structure. So if we can look at the molecular nature of those molecules and we see how they're folded up and how they work, we can also see how they're vulnerable, what the virus never changes, what it really, really needs. And that's where we target a drug or target an antibody.
So when you say see, what are you actually looking at?
We use very powerful electron microscopes, not a microscope, but a microscope. An 11-foot-tall microscope. The most powerful microscopes commercially available are focused here at La Jolla Institute on that coronavirus to understand the molecular nature of the atomic bonding by which that virus is built. If we can see it at that level, that is the complete roadmap that we need to understand how to defeat it.
So it's really quite a high-resolution picture or image.
It's extraordinary.
Wow.
Just down the hall is a SETI lab, featuring the work of Ullman-Saphir's colleague, Italian scientist Alessandro Sette. Liquid nitrogen canisters line the entryway. Patient specimens are kept in larger tanks under deep freeze so they remain viable for the myriad of tests this team will subject them to.
So your lab is one of many here at the center that's working on the novel coronavirus. Tell me about what you're doing there.
As you will know, reading in the news, the coronavirus infection, like most other virus or bacterial infections, sometimes has a bad outcome, a severe outcome. In other cases, the infection may be very mild, very unconsequential. So what we want to know is what's the difference? What tilts the balance one way or another?
What types of experiments can you do to help answer that question?
Well, we can measure the number of immune cells that see the virus. We can measure what do they do, what kind of things they do. Some white blood cells kill infected cells, and they do so by making a hole in an infected cell. We can look inside the cell and see what genes are up, what genes are down.
Sethi's team focuses on the human immune system. T cells are the body's line of defense against foreign attackers. Their job is to recognize and attack. But so far, COVID-19 has been able to attack seemingly at will. In Dr. Sethi's lab, they are trying to figure out why. Daniela Weitzkopf is a research assistant professor.
So we got a lot of boxes here. What's in here?
So all of these boxes contain peptides, which is small parts of the viruses, only 9 or 10 amino acids, and each of these boxes contains 96 tubes. Each of these tubes has an individual peptide in it. From each of these peptides, I know exactly from which protein in the virus it's from, and I know exactly which kind of people will be possibly able to recognize that, because they all based on different HLA phenotypes. So, which is different between you and I, we all have a different one. So, for example, when we go and test these, we will always make sure that we test the ones that are predicted for your phenotype in our cultures, in experiments.
You've covered the entire virus in just tiny 9 or 10 bits.
Exactly. The entire virus is chopped up in 9 and 10 mere bits, and this is necessary because, like, the T cells that are responsible to fight off the virus, kill the virus, they can only recognize very short parts of the virus. They cannot recognize the whole virus. They cannot even recognize proteins, like antibodies can detect proteins, but like the T cells can only see the very small parts. So that's what we exactly want to find out, what part of the virus can be recognized by the different kinds of people. And does that change depending on, if you have been vaccinated or you had natural infection, or does that change if you're young or old, or this is all kinds of questions we can address with that.
So when a human sample comes in, you would test that sample against all of these different possibilities and figure out which ones create the largest response?
Exactly, yes.
Down a staircase to a floor below is a room used by techs in the Crotty lab. This is where they are working on research involving vaccines.
I'm obsessed with vaccines and vaccine immunology, how they work.
Shane Crotty is professor here. In 2009, his research changed the way the body's antibody production and response is understood. Now he's focusing on COVID-19.
So the problem is, your body has 10 billion different cells. There's at least one of those cells that has a solution. And one cell is almost useless. And for that cell to be useful, it's got to divide, and probably divide to the point of being about 1 million cells. And obviously it's division. It's 1 going to 2, 2 going to 4, 4 going to 8. It takes a while. And so as a result, it's a while before you can make an antibody response that actually protects you. The whole point of a vaccine is to do exactly that, but before you ever get infected.
As the pandemic sweeps across the globe, from Asia to Europe, the Middle East, North and South America, and beyond, many believe a vaccine may be the only answer. Professor Crotty explained how that process is working right now.
One thing that a whole bunch of labs are trying to do right now with coronavirus is say, well, okay, probably people who are getting sick but recovering are making really good antibody responses. And so if we know what those antibodies are binding on coronavirus and what they're doing to coronavirus, that should accelerate vaccine development, because that gives you something to focus on. What's the right target? There are a bunch of possible targets, and even on the target, possible sites within the target. And the more we know about that, the more likely it is that somebody can make a vaccine that can mimic that. You want to take someone who was infected with coronavirus but got better, and take B cells from that person and find the right B cell that's actually able to kill coronavirus with antibody. For example, to do that, I would want to take 100 mils of blood from an infected person, actually a whole series of infected people. That's not a lot of blood. That's less than a normal blood donation. But that'll have, let's say, 100 million cells in it, okay?
It is a needle in a haystack.
Right, and so I'm interested in if I'm lucky, there are going to be 10 cells out of those 100 million that are the ones that I'm interested in. And probably it's more like one that's going to be the one that's actually really interesting in the end. So how do you find 1 in 100 million cells?
They do it with the help of these sophisticated machines using lasers which can peer into cells and make split-second decisions.
The flow cytometers are instruments, cell sorters are instruments that we use now, and they've gotten so sophisticated that we can sort something like 30 million cells per hour, one at a time, and we can interrogate each of those cells for 20 different parameters simultaneously, including, is it a B cell? So we can tell, is it a B cell or is it one of all the other types of cells in your blood? And then if it is a B cell, is it a B cell that can bind coronavirus?
Flow cytometers, like this one, are used to sort large mixed populations of cells into their individual cell types and components. Using lasers, cells are analyzed one by one, and then up to 14 different types of cells can be identified and separated. So basically, it's like if you had a bowl of chicken soup, you could put it into this machine, and the lasers would help tell you what's chicken, what's noodles, what's vegetables, and what's just broth.
Okay, so how does a vaccine work, right? There are about 28 licensed human vaccines. Almost all of them work on the basis of protective antibody responses. Antibodies are made by B cells, a cell of the immune system. Vaccines work on the basis of the immune system, right? So the crux of this is one of the genius things about the immune system is the immune system can make an antibody response to any pathogen that exists, any pathogen that ever has existed, and any pathogen that will exist in the future, right? So that's brilliant. That's
a superpower.
It is.
In the lab, the evidence is strong. SARS-CoV-2 should be a good candidate for a vaccine. There's plenty of proof the human immune system is working against the virus. People are getting sick, but they're also getting better. It's not like HIV or malaria. Neither disease can be cured by a human immune response. COVID-19 should. The question is, which vaccine will be the best solution?
So live attenuated vaccines are a classic approach where you take a pathogen and you essentially try to weaken it and develop a weakened mutant of it so that you've got the whole pathogen, your immune system can recognize it, and if the pathogen's already a pathogen that's going to elicit protective immunity, that's a pretty good path as long as you can weaken it appropriately. And so the measles vaccine is that way. The live polio vaccine is that way. The smallpox vaccine was that way. Those can be extremely effective vaccines. Or you can go with a killed approach, right, where you take the virus and your goal is to manufacture a very large amount of that virus and then chemically kill it so it still looks like the right virus, but it's now no longer infectious, and you use that as the vaccine. That's essentially what the annual flu vaccine is, and that's what one of the polio vaccines are. But the third and fourth are the ones that people are certainly pursuing the most with coronavirus out of the gate, so to speak, because they are ostensibly faster. So the third is a subunit vaccine, where you're just taking one piece of the bacteria or the virus and you produce that piece, a single protein or a single sugar. So I'd say the great example of that one would be the tetanus vaccine. If you can identify an antibody to coronavirus that does kill coronavirus, you can say, well, okay, what piece of coronavirus is that antibody binding to? And I just want to manufacture a whole bunch of that piece, and can I do so in a way that the immune system will recognize potently and make this kind of antibody. And a fourth category that people are very enthusiastic about is genetic vaccines. And so this would be immunizing you with some DNA or RNA that encode part of the pathogen so your immune system will respond to that. This is just a piece of DNA that would express part of the pathogen for a few days or a week or something and then be lost. These approaches have been really successful in animal models like mice. The most incredible appeal of both of those is that they are really fast and very cheap.
As the work goes on here in the Crotty Lab and in other labs like this one around the world, the odds begin to increase for finding the so-called needle in the haystack. It's a game of informed hunches and prolific testing that one day might pay off. The question is when.
So we know more, we can move faster, we have an expanded toolbox. How close are we to a COVID-19 vaccine for humans?
I think there is actually good reason for optimism. It's not an overnight process. It's not like what happens in the movies. It takes a lot of people and a lot of time and effort to puzzle it out.
But 1 year is different than 20.
Yeah.
Back upstairs in the Sete lab, they show us how they would run a test to record immune response.
So we're gonna open the box, and then we take off the cover of the peptides, because every single of these tubes, these are 96 tubes, so we can store 96 peptides at the same time, are all in here.
Each one is different.
Each one is different, exactly. So then we will use our 12-channel pipette. We're gonna take up the suspension, which is DMSO in this case. So this is to keep the peptide stable over time.
That's not water.
No. So once we have done this, we can close off the peptide box again. This can be in the box, and we can store it in the freezer. This is stored at -20. So all of these peptide boxes are labeled, categorized, so I will know forever where each individual peptide is. So then we can go to the freezer. This is our freezer where we store all kinds of peptides derived from all different viruses. And then we will just put our peptide here. This is from the SARS coronavirus. So we have all the peptides here. So this is all our yellow fever peptides. We have dengue peptides in here. We have Zika peptides in here. And then this is basically everything. Keep in mind, this is not the real viruses, right? These are just the peptides, which is why we can work so fast and we can work so quick.
We have technology in-house where we can get a blood sample from people that have not been infected. And so I could get a blood sample from you, and we can put those blood cells in the tissue culture system with pieces of a virus that we have synthetically made. And then we ask the question, in a population level, males, females, different ethnicities, different ages, what happens when they see the different pieces of the virus? So we can test a lot of stuff.
Finding the right combination takes time. The process itself is now part of their routine. The sample that will be tested is retrieved from the liquid nitrogen freeze.
So in this case, then I would go open the lid. And as you can see, I'm wearing gloves that protect me from the cold. And then I will pick the rack that I have been picking and lift it up. So all of these boxes have donor samples in there. And because we have a donor base where we keep track of them, I will know exactly in which box the sample of my interest is. Exactly.
We generate these pieces, and then we put them in the Petri dish, if you wish, or the test tube. And if a T cell sees that, they will go crazy. They will start trying to kill the cell, will secrete tumor necrosis factor or interferon gamma. They do all sorts of things because that's their job. They're like guard dogs, and they will see, they will go.
Now gives us an answer of what is actually recognized and what is not. So you can see here, these peptides have not been recognized versus these are strongly recognized here.
What's the difference between these bright pink ones and the darker circles?
So the brighter the color is, the more this peptide is recognized. So the brighter the picture here, the stronger the immune response against this particular peptide.
No one knows which will work best, but La Jolla Institute has activated an immense database of research which may hold important clues. The disease, COVID-19, is caused by the virus SARS-CoV-2. It's part of the coronavirus family that produced severe respiratory syndrome in 2003 and Middle East respiratory syndrome in 2013.
It so happens that SARS is the closest cousin of a new coronavirus. It's very, very, very similar. The proteins fold similarly. the structure of a virus is very, very similar, which means with high probability, those are the same ones that are going to be recognized in the coronavirus.
The hypothesis is that we would be able to treat this novel coronavirus similarly to the way that we treated SARS because it might behave the same way, or our bodies might behave the same way towards it.
Two viruses are always two different beasts, but there's going to be very large similarities.
When the novel coronavirus struck, the Sete lab hit the books. Their online search of scientific databases turned up a huge amount of pay dirt. 20,000 papers with 1 million so-called hits that suggested further investigation. Alba Grafoni conducted the research.
Tell me how this database works and how it can be a starting point when you're investigating a new disease.
Right. So one of the challenges when you have a novel virus is that there are not information available. But if you look into how the virus can be similar to previous viruses that already have been studied, maybe you might be able to find some portion of the virus that can be relevant for the immune response.
Her key finding? COVID-19 appears to be closer to SARS than MERS.
What we think this analysis is useful for is that it can kind of direct future scientists to actually do research on this virus on specific portion instead of looking at the entire virus. This is not a small virus. This is like 9,700 amino acids. So if you just try to do experiment without having an idea where to go, it's gonna take a lot of time for the scientists to actually get findings. So that's the main purpose of this.
It's a way of narrowing down where to look before you start experiments.
Exactly.
The race to find a cure is happening in labs like this one, around the United States and the world, all focused on solving this problem with more speed and more technology than ever before. That is where Professor Ullman Sapphire believes LJI can help.
So what we're going to do here at La Jolla Institute, what we've been asked to do, is to receive the different antibody molecules people are making around the world and actually compare them side by side and figure out, among the world's efforts, which ones are most effective and why they're most effective, and which ones we should combine. Because the right treatment might be 2 or 3 things combined together to attack the virus in 2 or 3 ways, right? What if the most effective treatment is 1 molecule discovered in Washington and 1 molecule discovered in Paris and 1 molecule discovered in California? We're not going to know that that's the right treatment until we have them all in the right place.
It's going to take time, compressed time. But still, a vaccine or a drug to slow the coronavirus isn't happening overnight. But I learned one thing. If you ask the scientific community, it's not a matter of if, but when this will happen.
These are solvable problems. So tackling other diseases that we all get, cancer, heart disease, diabetes, autoimmunity, these are really complex, and they're diseases of our own human molecules. Whereas these viruses are simpler. They're solvable problems. Lassa fever that we work on only has 4 molecules. Ebola, only 7. These viruses we work on just have a handful of targets, and their molecules aren't human. So you can target them and wipe them out usually without any toxicity to our own human body.